Hey everybody, what's going on? It's Alexander Williamson here with the secret history living inside your aquarium. You may already know that. This may be an insert to another video, but what I'm going to do is show you what I got under my microscope right now. Okay? Alright. So, on this slide that I have put together here, we have several kingdoms of life. We have algae and plants, we have cyanobacteria, and then we have the good bacteria colonies, which also have some fungi and other things in them that are forming aufwuchs, or positive slimy surface area made up of lots of organisms that uh, take nitrites, nitrates, ammonia, and they work with it. So basically, ammonia and nitrites, they turn into nitrates, as well as doing other things too. They turn lots of things into other things. It's the nature of what they do. They're decomposers and rearrangers of molecules. So pretty cool. Of elements, even. So we're not that zoomed in right now. Here we got your normal pain in the butt hair algae. So let's zoom in a good. Let's give it a let's blast off to 100 times multiplication. Zoom in here. So this stuff's getting kind of. This is that two kinds actually. You can see that purple, really stiff, stringy hair algae stuff that you get sometimes, and you can also see the cells and nerve center kind of things, even though they're not really nerves, but these nodules. Let me try to center one. Nodules on the string algae that allow it to break off and to split into multiple pieces, puts different cells and liquids and things in there, and it's kind of just like a little network rest stop or interchange spot. Also over here you can see the same kind of thing going on with more in a different kind of algae, but they all have super clear cell walls. You see these very specific lines in this stuff, and when we look at it, they're usually long, spindly cells. Let's zoom in all the way that I can do. This is 400 times. So this is your full zoom, and that is your algae. This is your hair algae, slightly drying out as we speak. But there you can see the different cells and stuff going on here. With it, there's also some detritus and other cells of fungi and protists and all sorts of stuff. But right there, you can see the long, spindly algae cells, even as they're drying up. All right, so let's pop back over to low magnification, and we see our hair algae. Let us pop over to the hero of the day, the sponge. So this is what sparked a lot of this work. This is a freshwater sponge. So, of the Spongilla order, or Spongilla, like Godzilla, I kind of like that. It looks like fiberglass, which is what it should look like. It has Gemmules making up its irregularities, which are like these little spines, and they get together and they form a dense clump. And also, you can see that the diatomaceous algae, that green algae, that's green spot algae, it gets together and it helps feed the the sponge. They work in tandem and they share chemicals. One swaps sugar for proteins and other ions, like I think there's one that gives calcium and one that does zinc also. But it's different for every species. So just take a look at that for a second and look at the dark hole in the center there, which is basically just a hole, and that is how the sponge it just sits in weights and it builds like a spider web and it catches all the stuff that floats by. So I'm going to kick it up, boom, a notch, where you can actually make out the cells in the sponge that are algae and not sponge. And here, take a look. This is the freshwater sponge. It looks like fiberglass. So take a look at that 1 sec. I need to change another setting. All right, so here we have, we're zoomed in over 100 times, and you can see the structure of the sponge. And this specific species is known as Ephydatia Muelleri, or Muelleri, whatever. I'm bad at pronouncing these sometimes. I never know what they want from me with all the eyes they put in place. All right, so let's zoom in like to the max. All I can give it. I can't give it any more juice. Let's see here. It's hard to get it in focus when we're this close. But we have a nice swath of sponge that I killed for you guys, unfortunately. But here you can see those cells which look like crystals, and that is notoriously sponge consistency. That's what you want to be looking for if you're trying to ID a sponge versus fungi. Fungi have little round spores everywhere, and they look different under here. I'll have to get some spores another day, but I couldn't find any underwater that I could grab super easily. So yeah, let's go over with the light. Let's flip the other light on, and let's look right here. Should be some. There we go. All right, so right there, those little nodules are called gemmules, or demules, is kind of what it sounds like when I hear other scientists talking about it. But the gemmules, they are not asexual. So the gemmules are like a male or female DNA packet, and they team up, float around within the same body, or float off into attach to other sponge things in the area. Sponge colonies, I guess is the word, organisms. And they can reproduce that way. Now, every so often, though, you get these black or dark, dense clusters like this guy, and you can see them forming too. Like over here, you can see how the crystals actually start forming by getting super dense, and the ends of those little spears that sometimes, the cells that look like spears, some of them, these ones are just tasked with catching things that float by and pulling them in, or I guess stuff gets stuck on it and then the cell grows around it. But the ones that have the little end on them, the little round ends on them, like this one in the center here, you could see they have either a female or a male gender, and they wrap it all up together, get smushed together as I understand it, and they end up in these little dark spots which can break off, and they are so protected, they grow a new type of cell layer, a stem cell layer around them, that they can now survive freezing, they can survive 120-degree weather in a hot spring. They're really cool, and these guys can then float off and awake when the time is right to build their colony. Now, a lot of people try to keep these sponges, and they do pop up quite a bit in the hobby, different types. This one's from all over North America and parts of Europe, but it's odd that it's in such a warm aquarium. So this is Ephydatia Muelleri, discovered in 1856 by a fella named Lieberkunz, Swedish dude. So that's what I've got to show you under the microscope here. Let me show you real quick so you get an idea that I'm not just pulling your leg or whatever. That I do have plant cells, not just algae. And when we zoom in on these, we end up seeing every little cell wall on the plant. You can see that they have cell walls that are thick with chloroplasts, little clumps of it. If we zoom in all the way to 400 times zoom, best I can do, you can actually see the little sacs of green chlorophyll in the plant. So, and it's hard because everything's a 3D object underneath the microscope, but that is what it looks like. So, pretty cool. Then there's also the main runway in between all that, and you get these veins that water and nutrients and things move through the plant with. But even when you're not that zoomed in, you can tell, oh, that is definitely a plant cell. But look at that, you see the dendritic patterning. Now, the last thing I'll show you before we hop back to looking at the sponge is, and I apologize if this is boring to some of you, but hopefully somebody will dig it. So this is from me. This is a blood sample. Now it's drying quickly. And I have an autoimmune disease, so my blood samples may be slightly different than yours. But for the most part, when we look at blood under the microscope, dried blood, let me reorient this real quick so we can see that. All right. Those are cells that are hardening, and they're all little circles, and as we get to the outside of this, you can actually see all the little circular disc cells. And as we zoom in all the way, you can see that that pattern is made up of, they look like little coins stacked on top of each other. It's really hard to get them to focus unless you have a slide cover on there. But you see the little round deals? Everything you're looking at here is stacks, like 30, 40, 50 tall, of the little round guys that have clumped together because they're, they're out of the body, and that's what they do with the hemoglobin. And the white blood cells have gone to the outside, sacrificing themselves. Here's some lone cells for you. So there's some white blood cells for you right there. And those are single human cell right there. So check that out. That's what's coursing through your body, my body. There's red and there's white. White look clear on here. Then lastly, we're going to hop over and look at bacteria. So this is good bacteria that turns the nitrates and or creates nitrates from nitrites and from other things. There is actual fungi mixed in with it here. There's like little spores and other doodads in here. There's even plant cells you can make out, like green plant cells and debris. But what this is it's my filter floss emptied out. There's a little egg. That's kind of cool. In here you'll find little nematodes. You'll find, these are all dried out now, but little organisms living in that complex bacteria colony. Right here you can actually see the bacteria building up, and its cells are so much teenier than human cells that basically when we're at the level that human cells look like the size of, I don't know, a pea on the screen or something, or one of those dark areas in the center there, you can see that their cells look so teeny. They still look like they're at the first level of magnification. So you can see some sponge cells there. You can see a little organism. Where'd it go? You can see This is algae that has died, those little perfectly square things, or they're plant cell walls that are broken off. There we go. That was a single, here's a little single cell organism there. But yeah, there's just an incredible world in here. And I just wanted to make it clear that once you have the microscope, you're not just making things up. You're actually seeing a whole world and able to, decide what the heck you're looking at. So that's what your filter floss looks like when it's scraped. And the sponge, dun 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 dun, dun. Where is the sponge? Looks like this, just so you guys know one more time. Pretty different, huh? Very interesting life forms. I implore you to get empowered and go learn about them. There's the diatomaceous algae that they like to live with. There's the little feelers and sensors that they have out on the edge, tells them where they're getting nutrients from and where to grow. The pocketing that goes on, like here's a pore, things get stuck in that channel there between all those little rigid things, and that orange there is actually food that it's eating, a little bit of something or other. And then here are two of those gemmules or spinules, I've also seen them called. But that is forming, it's a clump that gets denser and denser. And as I said, it can go out on its own, go dormant, and it has all the information for life for a sponge. And here is that more spongy look that we're accustomed to. If you see it here, let's pop back into the lowest magnification. And that looks more like a Brillo pad sponge right there with the, especially that little breakaway piece. So, all right, guys, I know that was kind of long and wordy. I apologize for that, but let's pop this off real quick. Okay, let's pop this off and let's show you guys what we were looking at. So what we were looking at is this stuff: blood, leaf, bacteria. There's the sponge right here with the diatoms on it, and the algae, the other kind of algae, very unofficially done, and then the bacteria. So that's what's going on there. I'll show you one last time the sponge. So I feel bad. Look at what we've done to this poor guy. I've sent out samples and all sorts of stuff. But there's the sponge growing down here. There's this little buddy here. And then there's this main sponge that we've kind of, I accidentally butchered it. I didn't mean to butcher it, but I slipped when I was taking a notch out, and it actually just like all peeled off. And some of it fell down below into the snail graveyard where the puffers eat, and some of it just kind of melted away over the next day. But then the last... check that out. The last sponge is right there, and it is feeding off the protein on this failed to win. This is losing snail eggs, but this same sponge has started with those little gemmules. They've started to drift, started a new colony, and they're eating that protein and num-nums that the snails left behind. These are snails that need the salt water to reproduce, neorites. And so same thing's going on here. Seems to be a trend, if I do say so myself. All right, guys. Well, the other thing I wanted to show you is that water that comes through here is very clear, for one. But for two, if we look at it carefully, you'll see little flakes of stuff float by. Let's see here. And everything floats by, and in this area, it comes whipping around the corner. The super strong currents up here, comes down, settles down through here, comes under all these branches and things, and then very slowly, almost in like a siphoning matter, it drifts through here. And I think this 1-inch gap between the glass and the stone that was taken out of a river probably had some of those spindules on it, or gemmules, and that is what turned into the start of this colony. But let's see here, can we see any of these little things floating past? Hold on one sec. I had some fish food, and it always shows the current. Maybe we can see it here. But there's actually a current in these little flaky guys that comes through. And here, you can see it coming around the corner really slowly. But I think that slow, continuous current is exactly what has allowed these sponges to take hold and enjoy this area. All right, guys, I hope you learned something. I hope you liked that. I know it was a little dry, a lot of dry. And I don't know. Let me know if you want to see other stuff under the microscope.